आइए अब हम बात करते हैं reagents और supplies की which are required for sequencing, DNA sequencing using sequenase enzyme. Buffers में deionized water is required, dithiotritol is required, DNTPs and DDNTPs of all four types. Four types of DNTPs and four types of DDNTPs are required at a solution concentration of 0.5 millimolar. Then we need the labeling mixture 5x for DDNTP and extension termination mixtures. Formamide loading buffers are required. For enzyme dilution, जैसा कि सीक्वेनेज एंजाइम है कंसनट्रेटेड फॉर्म में अवेलेबल होता है, इसको डाइल्यूट करने के लिए हमें डाइल्यूशन बफर की ज़रूरत पेश आएगी. दैट इज़ containing trace HCl, DTT, bovine serum albumin, and once prepared, it is stored at minus 20 degrees centigrade. 5 times sequenase reaction buffer containing magnesium chloride, that is the ionic solution, trace HCl, magnesium chloride, sodium chloride is required, and it is stored minus 20 degrees centigrade. Besides magnesium chloride, there is another option using manganese chloride. The composition is more or less similar except the molarity of manganese chloride. Here we are going to use 25 millimolar, whereas for magnesium chloride we used 100 millimolar concentration. Sequenase enzyme is required, and an optional enzyme, yeast inorganic pyrophosphatase, may be required. That is an option if you need to use it depending upon the experimental conditions. Oligonucleotide primer at a concentration of 0.5 picomolar in water or TE buffer. Then template DNase 1 microgram in TE buffer, that is trace EDTA buffer, pH 7.6. For labeling purpose, we need radioactive compounds that could be DATP with different variants like P32, sulfur, S35 or P33 labeled at different activities ranging from 1000 up to 3000. Special equipment में we need microfuge tubes or plates, whatever is the choice. Number of samples पे depend करता है. आपके samples कम हैं, you can use the tubes. आपके पास sample sequencing के ज़्यादा हैं, you can use 96 well microtiter plate. And the thermal cycler is required. Water baths are required, preheated at 37 degrees centigrade and 65 degrees centigrade. Centrifuge, normal microfuge rotor is required, which can centrifuge 0.5 milliliter of the tube or 1.5 milliliter of the tube. Normally this is called as bench top centrifuge. That table contains the volume composition of for 5x labeling buffer that is required for sequenase reactions. This side shows us the dideoxynucleotides. This one shows us the required DNTPs, and then one of each dideoxy. That is, we are going to use 16 units or 16 microliter of DCTP, TTP, ATP and GTP. Then other reagents includes sodium chloride 5 molar, 10 microliter in each of the 4 tubes, and water as per required volume. That makes a total volume of around 955 microliter or around 1 milliliter. All of these reagents, they are used as per suggested recommended volumes, and the remaining volume is made up to 1 milliliter by adding the 334 microliter of water. These volumes are adjustable. if you want to end up with 500 microliter of the total reaction volume, each and every volume has to be adjusted.